Thank you again for allowing me to speak. The invitation from Catherine was a welcome invitation. The second topic I was asked to speak on, and I broke it into a second section because it simply was too divergent from our first topic, is about haploid induction and its role in breeding. All right. Most of you are familiar with the concept now of doubled haploids and their role in a breeding program. And my intentions in this relatively short presentation is to walk you through the logistics of how these things work and why people are interested in them in many different crops at this point in time. So what is a haploid? Most of you are familiar that the traditional crops that we have are made up of either a diploid number of chromosomes, refer to them as diploid, or some form of polyploid, whether it be a disomic hexaploid or a autotetraploid, some form of that. Haploid plants basically possess the gametophytic level of chromosome, right, that a normal sporophytic version, the plant version, the whole plant version would have. And they represent the products of a meiotic event during meiosis when subdivisions occur. You get that haploid complement produced in that gametophytic phase. So whatever the chromosome number is of a normal plant species, the haploid will be half of that. So for example, in corn, we typically, from a cytogenetic standpoint, will say that corn as a normal sporophytic plant has a 2N value, which is equal to 2X, meaning that it is a diploid, and it has 20 chromosomes at that point in time. So a haploid of corn would be a designation of an N level of ploidy, half of normal, and that's X again, and that would have 10 chromosomes. If we talk about a polyploid such as hexaploid wheat, and we look at the same complement, we would have 2N equals 6X, representing the three different genomes that are present there, or excess copies that we have in this disomic polyploid, with 42 chromosomes. So a haploid in a wheat situation would actually be an N value, again, N referring to the haploid nature, the gametic phase, with a 3X number of copies of the genome and 21 chromosomes, one set from each of the three genomes. As you know, wheat has an A, B, and D genome. So it possesses a full complement of the genome, but it's in a hemizygous state, meaning that there's only one allele per locus, because there's only one set of chromosomes in there, all right? Phenotypically, haploid progeny are smaller, usually, and slower to develop, and most often they're completely male sterile and female sterile. We've known about haploids for over almost 100 years now. The first report was in Jimson wheat back in 1922. Since then, they've been found in most every plant species that we have looked for them extensively. So they're not uncommon, but their frequency is generally not high unless factors or modifications have been made to increase their frequency. Now, key to this haploid nature is that if we're going to use haploids, we have to have a way to double them. And so I say right away that chromosome doubling is key to this process, because remember, we don't talk about haploids in a breeding program. We talk about doubled haploids. And doubled refers to the fact that in order to make these haploids useful, we have to double the chromosomes. We have to go from this 21 chromosome with one allele per locus to a 42 with two alleles per locus. And this is the appeal of a double haploid. If you start with only one allele per locus and you double that, by definition, you are going to complete a completely inbred line, and it should restore fertility to that plant. And if you can produce an inbred line in one generation, contrast that to self-pollination for several generations to complete that. It saves time. So how are haploids produced? Well, it depends. Just like in so many ways, the biology of the crop dictates to a certain extent the things that can and cannot happen. But basically, there are four methods of producing haploids at this point in time. The two that have been around the longest and known the most about are gynogenesis and androgenesis. These are basically taking the ovary or the flower in the case of gynogenesis and extracting out the haploid gametophytes, the ovules, the cells within the ovule that are haploid, and regenerating plants from those particular floral structures. It's from the female side. And there are examples in the literature from sunflower, safflower, flax, and several other crops that have done this. Androgenesis is anther or microsporogenesis pollen culture of taking those haploid pollen cells and gametes in the pollen and regenerating progeny from them. And there are numerous crops, I've just listed a few here, with reports of application of this type of approach. Both of these rely completely on pollen, a laboratory step phase of regeneration, of plating and putting them on agar and regrowing plantlets that are derived therefrom. More recently, in the last 30 to 40 years, the concept of white hybridization with subsequent chromosome elimination has occurred. And what we mean by this, and it works quite well in many of our cereal grains, especially the small grains, you take a wheat plant or a barley plant. In barley, you can actually use a related species of hordeum. Wheat and oat actually rely primarily on maize pollen. And you pollinate wheat or oat with maize pollen, and the corn chromosomes, when wheat is used as the female, are eliminated as that embryo develops, and so you're left with a haploid complement of the respective wheat or barley that can be recovered, grown in tissue culture, or recover the embryo through embryo rescue, and then doubled to produce a double haploid. Finally, the most recent to be applied, although it's been known even for longer than white hybridization, is the concept of genetically controlled haploid induction systems that create haploid progeny from seed. These are wonderful in the sense that you never have to go through a tissue culture stage, and they're applicable directly in the field level. They're controlled. Corn is the classic example, and the one which was first described back in the late 1950s, and then really came on board about 10 to 15 years ago as a true system of extracting double haploids in corn after 40 years of breeding to increase the frequency of this genetic control that affects the induction of maternal haploids based on a paternal pollinator. And the gene most commonly associated with this is called matrilineal. Although it's not completely penetrant, there are many other factors that influence the induction frequency. I'm going to show you some examples of the latter two. You can look up the gynogenesis, androgenesis. There are reports out there, well documented. But for purposes of that, before we go into that, I want to talk about the factors that influence the frequency of recovery of haploids, because these are really important. If your frequency of recovery or the cost of recovering is so expensive that it's simply easier to use traditional approaches, that's what people will do. Those are influenced by several different several different factors depending on the type of regeneration induction you're using. For example, almost all of them have some level of genotypic specificity that has to be overcome in order to be truly applicable across a crop species. Cultural methods, tissue culture methods, and genotype response to that are important. The ability to regenerate through tissue culture can be really important for gyno or androgenesis. Amenity to doubling chromosomes through spontaneous or chemical means, i.e. colchicine, are really important. If you can't double them, it's hard to use them. And then finally, for the induction system in corn, the frequency at which induction occurs becomes really important. If you're very low, it takes large numbers of seed in order to get and identify haploids, and methods to identify those haploids as well. For example, corn started out inducing, sources of inducers of corn started out at very low frequency of induction in the less than 3-4%. They have improved those drastically with selection over the years to where it's not uncommon to hear people say that 25% of the progeny from this method are expected to be haploid. In sorghum, we've found inducers as well. However, the frequency is still very low in the 1-2% range, which makes it difficult to utilize the technology at that low frequency of induction. So I'm going to walk through haploids created through white hybridization. This is a schematic taken from Humphreys and Knox. Please go and read that, and with all due credit to them for this. Basically, what you're doing with this, and we're using wheat here as the example, you have emasculation, all right, to remove and eliminate self-pollination. Once that is done, you pollinate that plant with corn pollen, okay, onto that. A auxin to sustain embryo development is applied to that pollinated panicle, and about 2 to 3 weeks after that pollination, they are harvested through embryo rescue. That haploid embryo is removed and put in culture in a nutrient medium to generate haploid plantlets, which then are treated with colchicine and doubled, and then a fully fertile panicle produced from that plant manifests seed that is completely inbred and ready for evaluation. So the process here, if it's a winter wheat, you may have to vernalize. It could take up to 9 months or a year to get this done. But relatively speaking, to go from a F1 breeding cross to an inbred line in a year is relatively efficient compared to selfing for 5 or 6 generations. This is another source that you can look online and see this reference here from Santra et al. That puts it in more physical pictures where you have an emasculated panicle, corn pollen, you pollinate, put the auxin inducer in there, let those grow for 16 days, recover those, extract the embryos, plate them, and then colchicine, and then let them grow, colchicine double, and eventually you produce a spike with fully set seed that is completely inbred. Corn and haploid induction systems are much more field oriented in their application. And here, this is taken from Chaikum in a recent publication in Theoretical and Applied Genetics, where you see the inducer line pollinating all of these D-tassel breeding crosses, F1 breeding crosses. And so you see the detail here that they're putting that inducer line on this kernel here. And then you simply take that seed, their inducer line has a embryo or endosperm specific color marker in it, so that if you have a haploid develop, you see that in the progeny. And you see right here that you see full color penetrance in these diploids. In the haploids, you do not see that full color penetrance. And so using color, you can isolate putative haploids from the primary source that you have in here. And this is extremely valuable. If your induction rate is 10 to 15%, it means you've eliminated 90% of the seed that you might have to screen and gotten to a much higher concentration of haploids in the progeny that you screen, which are then germinated. Okay, they clip the coleoptile, in this case, the tip of it, to expose the growing point, in which they then take all of those for a particular population and they treat them with colchicine and then distribute them into a flat and eventually transplant those into field conditions. Okay, and most of them they hope will be fertile and produce some number of seeds. Now, these don't look near as good as that wheat hybrid or that wheat inbred line did, but remember, corn is an outcrossing crop that suffers from inbreeding depression. So you see some of that manifested in these double haploids that are recovered, but they are now inbred lines and they're applicable to a hybrid breeding program. So what does this get you? Well, this is a schematic from the Syngenta program that they use to explain doubled haploids. I borrowed it from them. And it shows very simply the manifest difference here, that if they look at this and to get to an inbred line requires 7 generations, depending on whether that's done 2 generations a year, that means 3.5 years. Whereas here they go from F1 segregation to an inbred line in 2 generations. So it saves significant amounts of time. In the day and age in which we live now with genomic selection technologies, this is where the true value of the technology resides. And I give you this schematic because it's reflective of our own program in sorghum and our interest in trying to develop the haploid system, because it shows you timeline differences in here. This is our traditional approach right here. And you'll note that to get to hybrid evaluation of anything in our program takes a minimum of 4 years. From the start, from the original cross through multiple fall and winter seasons to get to hybrid evaluation in what we would call phase 1, the first time we've seen test cross hybrids. If we use double haploids, we can use double haploids and we can have inbreds by the end of the first winter year, so 1.5 years after we start this process. And we could actually test cross those because remember, sorghum is not subjected to the same level of inbreeding depression that corn is. We could have hybrids in the field in the second year, 2 years after we made the original cross, and then we could advance and move forward with that. In addition, then, why this becomes even more important rather than just the speed is if we add genomic selection to this, because once we have an inbred, we have a repeatable genotype, and that genotype can be used in genomic prediction models to identify the top producing hybrids. So we could use that, like they're doing in corn, to eliminate 80% of these haploids from evaluation in field trials and predict their first observation or phase 1 evaluation performance based on genomic selection models, which allows us to focus on what we would consider phase 2 hybrid evaluation. So not only are we going 2 years faster here, if you'll note, to get to phase 2 hybrid evaluation, we're going 3 years faster when you combine haploids and genomic selection. And that's why these two systems, genomic selection, which others are talking about, and doubled haploids, are so co-linked with each other because they synergistically improve the efficiency of the breeding program by allowing you to produce inbred lines quickly and then predict their performance and move on to advanced phase testing very quickly. So they're going to reduce that cycle time. They're going to match well with genomic selection. And you see them used if you can extract numbers sufficient to make it feasible in a breeding program. So, in summary here, they produce highly inbred lines in one generation, saves time. Highly amenable to the genomic selection methodology because they eliminate those medium phase where segregation is still occurring. On the negative side, and this can be a positive or a negative, it's simply a statement of fact, it will limit genetic recombination in your breeding crosses because remember, when we make a breeding cross, we have parent 1 by parent 2, we get an F1, and we are going to cross that inducer line right here. And we're going to get a haploid right here. And then we're going to double that. When we do that, the only time meiosis allows for recombination is right here, one time. So you get one shot at recombination in that. So if you have linkage blocks that you want to keep together, then this is fantastic. If, however, you're trying to break up some linkage blocks and break the association between a negative and a positive trait, you may need more recombination. Hence, those extra generations of selfing or random mating may be important to break up those linkages before you induce double haploids into that system. So that time is important in that situation. We're talking about a technology now that you see commonly used in many of our major crops. There are other crops that would definitely adopt these systems if their systems can be fully developed, and they're simply not at this point in time. Ultimately, their use is a combination of logistical feasibility versus relative benefit. They are very labor intensive. They're very unlikely to be used in systems where there is not the resources available to do it because they are very amenable to large scale and intensive production with some infrastructure. So you have to assess that on a case by case basis, crop, situation, target, goals of a breeding program to determine if double haploids are going to be useful in that system. If there are questions, I'll be happy to answer them during our discussion session coming up next week. Thank you for your time.